Hello, this video is about an organic lab 1 experiment, isolation of chlorophyll and carotenoid pigments from spinach. I guess most of us learned spinach first from papaya cartoon at our early ages. Whenever papaya feels the need for power, he squeezes open a can of spinach, pours the greens down his throat. Why did papaya eat so much spinach? The secret power of spinach was attributed its high iron content, which is not true. The spinach supermeat was created by chemist Erich von Wolff in 1870. He accidentally misplaced a decimal point while recording the amount of iron within spinach and changed the iron content from 3.5 milligrams to 35 milligrams in a 100-gram serving. This legendary iron content was corrected 67 years later. Iron content of spinach was a myth, but same cannot be spoken for its chlorophyll content. Although every plant-based food that's naturally green in color has some amount of chlorophyll, spinach is a standout with its much higher chlorophyll levels than others. It contains about 24 milligrams of chlorophyll per cup, while most of other green vegetables contain 4 to 50 milligrams per cup. Due to its high chlorophyll content, spinach has deep green color. The structure of chlorophyll consists of close-ring tetrapearls chelated with magnesium atom in the center. Hence, is a metal complex pigment. Carbon-oxygen and carbon-nitrogen bonds and also magnesium bonded to nitrogen leads chlorophyll to be polar molecule that is almost ionic. Chlorophyll consists of two parts. A porphyrin nucleus containing nitrogen nucleus similar to the hemoglobin in our blood, but containing magnesium rather than iron, and a hydrophobic tail with 1 carbon atoms, named as phytol. Phytol attached to the porphyrin ring makes chlorophyll fat-soluble and insoluble in water. All types of chlorophyll share the magnesium porphyrin-like structure. Chlorophyll occurs in several distinct forms. This diversity is provided by the different groups bonded to the carbons at X and Y positions. Two of them, chlorophyll A and chlorophyll B, are of particular interest in food coloration because they are common in green plant tissues. They only differ slightly. Chlorophyll A has a methyl group in a position Y, where chlorophyll B has an aldehyde. The different side groups in two chlorophylls change the absorption spectrum to slightly different wavelengths. When spinach is cooked up, the chlorophyll starts to break down. The side chains may fall off some of the chlorophyll molecules, making them water-soluble, so a green color leaches out into the liquid. Some acids are released as the plant cells break down. Due to heat and acid exposure, the magnesium gets removed from the center of its ring structure and replaced by an atom of hydrogen. The color of vegetables changes from bright green to olive gray. This results in transition from chlorophyll to pheophytin. Chlorophyll A gets turned into a molecule called pheophytin A, and the chlorophyll B gets turned into pheophytin B. Besides chlorophyll, there are several types of pigments that plants produce to maintain their vital functions. The intense production of chlorophyll due to photosynthesis activity overpowers other pigments' expression, although they are naturally present all year round in the leaves as well. However, in the fall, as a result of decrease in sunlight, chlorophyll rapidly begins to decompose, allowing other pigments to be visible. Carotenoids, together with chlorophyll, form the main two categories of these pigments. Carotenoids are yellow to orange or red pigments derived from isoprene. Isoprene serves as a building block with its 5-carbonate structure for more than 700 different types of carotenoids. There are two broad classifications of carotenoids: keratins and xanthophylls. Keratins are hydrocarbons. Alpha, beta, and gamma keratins are the main types of keratins and precursors of vitamin A. Beta-carotene differs from alpha-carotene by the position of one double bond on ring structure. Gamma-carotene differs from both by having the ring open, of which the position of double bond changes in alpha and beta-carotene. Xanthophylls, the precursor of plant hormones, are yellow pigments. They are a subclass of carotenoids and contain oxygen in different forms like methoxy, hydroxy, or carboxy. Therefore, they are polar. For example, lutein, one of the major xanthophyll, contains hydroxyl group. Photosynthesis in plants and algae takes place in chloroplasts. Thylakoid membrane in this organelle is the specific part in where the light-gathering complexes are embedded. Chlorophylls are the most common pigments, however other pigments such as carotenoids are also present. Within a photosystem, many chlorophyll molecules absorb light and the energy of excited electrons is passed from one molecule to another, funneling the energy into a reaction center. Minor pigments also transfer the energy they absorb to chlorophyll A, so that the energy can then be used in photosynthesis. It is proven that chlorophyll and beta-carotene work together to maximize photon absorption to increase the rate of synthesis. We'll continue with the technical methods used in the experiment. Since we have already watched the principles of thin layer chromatography in another video, here we will only explain the other technical method, named the column chromatography. Column chromatography is a purification technique involving the separation of compounds by the same mechanism as other chromatographic techniques like TLC. It is used to separate a mixture by distributing its components between a stationary and a mobile phase. It is classified according to the physical states of stationary and mobile phases like liquid-solid chromatography, which we will use in our experiment. Now, let's look at some terms used in column chromatography. Stationary phase remains fixed in place, acts as a brake on most components due to its polarity and good adsorption properties. Mobile phase or eluent is made of solvents and flows through the column by dragging out the components in the sample. Column is typically a glass tube into which materials are loaded and packed. The components with lower adsorption and affinity to the stationary phase travel faster. The components that move fast are removed first, whereas the components that move slowly are eluted out last from the column. The first step of running a column is choosing the right column. You can use differently sized columns to separate samples in amounts from micrograms to kilograms. While the diameter of column is directly related to the scale of your sample, its length depends on the difficulty of separation. Easy separation results from components differing greatly in polarity. The more difficult the separation, the more adsorbent will be needed, which means a longer column is required. The second step is picking a stationary phase. Alumina and silica gel are the most common adsorbents used as stationary phase. They vary in activity and particle size. The adsorbent is very active if it has a great attraction for solute molecules such as alumina. Silica is less active and a good all-purpose adsorbent. Since it has silicon, oxygen, hydrogen bonds at the surface, it is very polar. In the table below to the left, we can see which functional groups adsorb strongly on polar adsorbents, and in the big figure to the right, we see the possible interactions of compounds with alumina. Particle size of adsorbents affects the flow rate. The smaller the particle size of adsorbent, the slower the flow rate. On the other hand, if particle size is too large, then it ends up with a poor separation. For a good separation, column must be packed properly. For example, if it has an uneven surface or an air bubble stuck inside, channel formation might be seen. In this case, column chromatography becomes useless. Also, never let position your column like pizza tower. Make sure that you clamp it perfectly vertical. Otherwise, the slanted bands you get will have no effect on the separation, even though you correct the position of the column during the process. Two types of packing are applied, dry packing and slurry packing. In dry packing, you add your dry solid phase into the column. In slurry packing, you mix both liquid and solid outside of the column and pour it into the column. Not to cause any channels, once you wet the solid phase, you must keep it moistened all the time by covering the top with the solvent. Preparing the eluent comes as next step. What an eluent does in column chromatography can be summarized with one word: transportation. It transports components of the sample along the column after removing them from the surface of adsorbent. You can visualize it like Element is the bus, and the components are the passengers waiting at the bus stops to be picked. For sure, adsorbent is the bus stop in this case. Once the silica is well packed, the mobile phase element is added until it reaches the top of the silica. If you pick a polar solvent, it may displace with the solute molecules and become adsorbed itself. For this reason, you must start with a solvent of low polarity and increase the polarity gradually by changing the mixture of solvents. The last step before running the column is loading it with the sample. Do not load your sample until you have all your materials set up. Only when you are ready to run the column, load your sample gently to the top of saturated silica. A layer of sand is then added onto the sample to ensure it is not disturbed by the addition of more eluent. The eluent is then continuously added to the system until the mixture has been separated. Make sure the eluent level never drops below the top of the silica. If you need to change the eluent during process, first let the previous eluent drain to the top of the sand. Then add the next one. As the eluent is flushed down the column, molecules will come off the column at different times. Relatively less polar components that do not hold on tightly to the stationary phase will elute from the column more quickly and can be collected as fractions. In case that components are colored, change collectors each time a new band of solid begins to come off the column.